This method can help answer key questions in the field of alternative pre-mRNA splicing. Specifically, it provides a robustly sensitive assay system to examine alternative splicing patterns in situ in tissue slices. With this method, alternative splicing can be analyzed simultaneously in many cell types that are contained in complex biological structures such as a mouse brain.
The main advantage of this technique is that it uses short antisense exon junction probes in RNA in situ hybridization to produce isoform-specific hybridization signals. A built-in signal amplification technology increases the sensitivity of the detection to provide robust hybridization signals. The signals detected with exon inclusion and skipping specific probes are then used to calculate the percentage splice-in values in different anatomical areas of a mouse brain. In this study, NF1 exon 23A is used as an example to illustrate the use of the assay system. However, the system can be adapted to analysis expression patterns of many alternative exons.
To start mouse brain section deparaffinization, fill 2 washing dishes with 250 milliliters of xylene and another 2 with 250 milliliters of 100% ethanol inside a fume hood. Insert the tissue slides into a washing rack. Next, place the washing rack in a xylene-containing dish and incubate for 5 minutes while moving the rack up and down.